Welcome everyone to skeletal muscle fatigue and post-exertional malaise in patients with long COVID and implications for ME CFS. I'm the former president and CEO of Solve M.E. Delighted to be with you today, and very excited. Today we would like to welcome Dr. Rob Wüst, who's an assistant professor at the Free University in Amsterdam. And he's working in the area of human movement and metabolic physiology. Rob has over 10 years of experience in exercise and cardiovascular field, including expertise in oxygen consumption, specifically the mitochondrial function and cardiac and skeletal muscle function. Rob performs his research at the VU in Amsterdam in the field of cardiac function, inflammation, and metabolism. And he also teaches at the Amsterdam University College. So in 2022, Rob was a recipient of the Solve Ramsay Research Grant. I was still there at the time. And so I'm really excited to now go full circle and see the results that Rob and his team were able to get in this time frame. And specifically, the grant was for microclots, skeletal muscle pain, and post-exertional malaise in patients with long COVID, from pathophysiology to treatment. And we'll hear today mostly about the pathophysiology, and Dr. Wüst will share with us some of his thoughts about potential treatment and future research directions. So very excited to have you with us today. Dr. Wüst, please go ahead.
Thank you very much, Ovid, for your nice introduction. And I'll start sharing my screens. You should be able to see my screen if I'm not mistaken. Correct? Just.
Yes.
So thank you very much for the introduction, and I'm happy to give you an update about our work on that we perform on long COVID patients, but also implications indeed for ME CFS. And we particularly focus on skeletal muscle function because that's our expertise as muscle physiologists and exercise physiologists. And I really would like to highlight something about more about the pathophysiology of particularly post-exertional malaise that we'll talk about in more detail. Before I dive into the details of this project, I really need to thank the people that were part of this project and have led me on the way essentially through until here. The project started off with a clinical question from one of the post-COVID clinicians in our local hospital, asking, so these people are fatigued, that we see are fatigued, they cannot perform exercise anymore, and they worsen even with when we send them to the physiotherapist. There must be something wrong in their skeletal muscle, in their body, because we really don't think this is a psychological problem. So let's dive together into this problem. And that's how we did. It was a deep dive, and with the help of Solve M.E, we're able to keep the initial students who worked on this project. So Braden Charlton, he's a Canadian, and he as his master project, he worked on this project, and now does a PhD with us. Simply based without the help of Solve M.E, we would not have been able to keep him for longer. So that was actually the starting point of this whole project. So I'm really grateful that Solve M.E quite early on realized that this project may be important. So thanks for that. Important background information, I think, that is relevant for this talk is that patients with long COVID as well as ME CFS suffer particularly from fatigue, brain fog, dizziness. But an important parameter is the post-exertional malaise. And if we look at the current hypotheses in the field of long COVID and ME CFS, we don't really understand what this symptom really does and why it occurs. People have suggested changes in the immune system, autonomic nervous system too, circulation, the microclots. We'll come to that in a minute as well. But little focus has been placed on the skeletal muscle system and changes that occur that contribute there to the symptoms that we see in these patients. Important to realize is that the number two of the two most important symptoms that people suffer from when they suffer from long COVID or ME/CFS are both fatigue and post-exertional malaise. And some people see a lot of overlap between the two feelings, that people are more fatigued after exercise, but any person who does exercise will be fatigued for some time after extensive or exertional exercise. But there's something else essentially. So these things are really two different phenomenon. And that I'll try to highlight in the next half an hour or so. So post-exertional malaise is not just fatigue. It's the worsening of general symptoms or the development of new symptoms that are generally happening within 48 hours after physical, cognitive, or mental exertion. It doesn't have to be only physical. It can also cognitive or mental exertion that patients suffer from that triggers a crash. We don't know that the threshold above this occurs. We do know that this is patient and time specific. We don't know the underlying pathophysiology of those time specificity, and why some patients have a very low threshold and others a slightly higher threshold. And it can also be that physical, cognitive, and mental exertion have different thresholds. And that's again something that we don't really understand in a lot of details. But we do know that this PEM, post-exertional malaise, I'll refer this to PEM, that can last for days or weeks, or maybe even set back recovery for more than months. And that's actually something that as an exercise physiologist is really strange to me as when I started looking into this a few years ago, because I always teach my students exercise is good for you. And this, interestingly, has overlapping with other post-viral and post-acute infectious disease, such as ME/CFS. So when I teach my students exercise physiology, we always tell them exercise is good for you, exercise is medicine. But in this condition, in these diseases, it's exactly the opposite. And that's actually, from a scientific point of view, a very interesting factor, because we don't know much why that is that patients with long COVID and ME/CFS are suffering from this particular symptom, and what we can do about this. So it may well be that one day you look perfectly fine and you look healthy and there's nothing wrong with you. That sometimes happens when you would see a clinician or a physician, but the next day that's where the crashes come in, and that's actually when the real disease is showing up. And unfortunately, that's also the moment when the doctors are back at work again and seeing other patients. So this is something that is very difficult to highlight essentially to physicians and to GPs, that you may look perfectly fine when you see the doctor, but the next day that's when the crash is coming into place, and that's causing a real problem. Unfortunately, also, we don't have clear diagnostic criteria for the disease, and that makes it also difficult to really specifically say you suffer from PEM and you don't, and that makes it also very difficult for clinicians on the left-hand side to diagnose patients based on just seeing the symptoms, seeing the participants at that time point. So we dove into this, and we published a paper recently about this phenomenon, that post-exertional malaise, and we focus specifically on skeletal muscle changes. We're super grateful with all the work that all the people that have been done over where the colleagues of us have contributed on, and let me just dive into a little bit more what we exactly did and what we found on a more patient level. But first of all, the important thing is that post-exertional malaise is normally diagnosed based on a 2-day exercise test. Now, a 2-day exercise test is actually is double trouble, so to say, because the first exercise test is there to induce the PEM, and that can already set back recovery as we just saw. But the next day, the patients are asked back into the hospital to do a second maximal exercise test to see if they reach the same level. If they don't reach the same level, then the diagnosis is, yes, you have post-exertional malaise. But that means that the patient has undergone 2 times of an exercise test, and that will set back their recovery maybe even longer than just a single exercise test. So we thought that may not be the right approach to study this. Instead, what we opted for was a 2-day muscle biopsy test. So we took muscle biopsies before an exercise test, and we also took blood and questionnaires. And then we did this cardiopulmonary exercise test to study not only the whole body exercise tolerance or the capacity of the patients, but also to induce this PEM. And the next day we asked them back, mainly with someone who could support them to give another muscle biopsies. And these clinicians, they were really instrumental in this project, because without their help and their free time, essentially, they mainly did it in between other surgeries, and on the 3 days they came to the hospital to take the biopsies for our study. And then we follow these people up for up to 7 days, but often actually much longer too, to see how the recovery went and if they are recovering from this PEM induction. Importantly, the patients were diagnosed with long COVID based on interviews and medical history. So we knew that they had long COVID, and the inclusion criteria that they had to have post-exertional malaise. Without that specific symptom, we would not include patients. And that means that sometimes we get criticism that not all long COVID patients are suffering from the change that we see in the next slides, but that's maybe because they don't suffer from this particular inclusion criteria, post-exertional malaise. And they had to have a minimum of 3 months of persisting symptoms. We do know that exercise and skeletal muscle changes occur after hospitalization or after severe COVID infection. So what we wanted to avoid was that the hospitalization itself or other comorbidities could influence our results. So we really went for a very homogeneous group of patients that had specific symptoms related to the long COVID and not to hospitalization or post-hospital recovery problems or other comorbidities. Here you see some changes in the or some differences in groups, or actually they're very similar across the groups in years, the weights and the heights. And the working hours was very clearly different between the 2 groups. Importantly, that's something that will come up in one of the later slides, is that the average amount of steps per day was significantly lower in the patient population in red compared to the controls. However, you also see that there are some controls that are relatively inactive here on the left-hand side, and some other patients that were actually relatively active compared to the average or the lower end of the participants or the healthy participants. Also, we checked this compared to normal daily activity levels, and we spotted that the average level of the activity levels in the patients was very similar to the normal healthy American. It's maybe something to realize for later. So our healthy Dutch people were a little bit more active than the average American, which also is in the same range as our patients. But of course, they don't suffer from PEM. So what did we find? Is we first looked at the VO2 max. The VO2 max is a marker for exercise capacity. And you see it on the left-hand side. You see the patients always in red and the controls, healthy controls in white on the left-hand side. And we saw indeed a significant difference in the maximum exercise capacity between patients and controls. And that's actually something that we would expect. However, we also spotted that some patients actually still had a relatively high maximum oxygen uptake consumption during these maximal testing. And those were mainly like bodybuilders or professional athletes that we also included in our study cohort. But of course, they also had these persisting problems and PEM. When we look deeper in the skeletal muscle, Skeletal muscle can be made up of white and red blood fibers, and the red fibers are very oxidative and are able to generate energy for a long period of time and make you, for instance, run a marathon, allow you to run a marathon. On the other hand, the type 2X fibers, those are the glycolytic fibers or the white fibers, and as you can imagine. For instance, those cannot produce energy for a long period of time. You can also compare that to a duck breast versus a chicken wings. The chicken wings have very white meat, whereas the duck breast has very red meat, and that's because a duck can fly and a chicken cannot. What we spotted was that the patients here in red again, they had lower percentages of the very oxidative fibers, so the red fibers, whereas their muscles were had more white muscle fiber types, the low oxidative fatigable fibers. So this can directly link to their feelings of fatigue. At the same muscle size, we also spotted that patients were able to produce less power on the bicycle compared to healthy controls, which were in the which produce more force for a given muscle size. So in this relationship, we see that patients are not able to generate the same amount of force that you would expect based on a healthy population. And lastly, what we observed was that a marker for mitochondrial function or mitochondrial density, which are the organelles to produce energy and produce are able to withstand fatigue. In healthy controls, we see this nice relationship between the amount of mitochondria that you have versus the exercise capacity on the bicycle. However, in the patients, this relationship was completely lost. And that made us think that maybe there's something wrong with their energy producing pathways. Here you see an image of some of these organelles that we focus on. They are these mitochondria that are. This is a zoomed in image with a very high magnification inside the skeletal muscle, and we can visualize these mitochondria using specific microscopes, and we can look at the structure of the muscle. Here you see the lines of the muscle contractions, and here you see in the healthy participants very nice these mitochondria, these organelles that are producing all the energy for the muscle contractions. What we found was that the oxidative phosphorylation capacity, or the ability for these mitochondria to produce energy, was already significantly lower before we performed this PEM induction, whereas the density itself was not so much different here in B. However, this indicates that there is an intrinsic problem with the ability to produce energy from these mitochondria. We also spotted that after the exercise, in particular in the patients, but also a little bit in the controls, we saw a significant reduction in the energy production pathways, but it was very markedly becoming different in the mitochondrial density between the two groups. So when we looked at the metabolomics, so the metabolite concentrations at rest of these muscles, we spotted that the patients had more metabolites related to the glycolysis, the glycolytic pathways that ultimately produce lactate, and less of the metabolites related to ATP production, ATP itself, but also the ATP production in the mitochondria. So we think there is really an altered metabolism occurring in patients that can underlie their fatigue. And what we also saw is that some of these markers change rapidly upon the induction of PEM. So this is worsened, the metabolites worsened upon induction of PEM. So back then there was a very easy explanation, we thought, because there was this theory from Dr. Pretorius from South Africa that microclots would underlie all these changes that we see. And what we spotted was indeed that these microclots were present, and the idea was that the theory was that these microclots in the blood, as you see here on the left-hand side, they're able to clot the blood flow to the skeletal muscle. And the idea was then, okay, after this clot, there is less blood flow, and there's less oxygen delivery and nutrient delivery, and therefore these muscles are becoming more fatigued. That's super simple explanation, and we thought, okay, this is going to be a simple test. Let's see if we can find those clots in our skeletal muscle. So with the help of the Solve project that we started, we were able to visualize these amyloid-containing deposits, we call them now, in skeletal muscle. And indeed, we did find them in the skeletal muscle. They were also higher in the patients versus the controls. I see that the image itself is not so nice anymore, but there is a difference here in the red versus the white. At baseline, the patients had more these microclots compared to our healthy controls. And also there was an increase upon exercise. So we also think that maybe, particularly in the patients, we saw some patients had a really big increase in these amyloid-containing deposits. So we thought maybe there is something happening, but let's see if they are then inside the capillaries. So here you see in purple the cell membrane of the skeletal muscle. You see a vessel here. With patients versus a healthy control. And in green, you see the microclots containing here. If you zoom in a little bit and also stain for the endothelium or the blood vessels here in pink, and now the cell membrane is in blue, you see that the green dots are actually not inside any of the capillaries. We couldn't find any single microclot inside a capillary, but they were between the fibers, but we just could not relate any of this theory to blocking of capillaries. Then we thought maybe they're in the lymphatic vessels that are draining the muscle and are linked to the immune system, but also there was no sign that these microclots were able to block the lymphatic vessels. So we really had to dive deeper into this, and currently we are continuing our work with Dr. Pretorius to also see what these clots are in the blood and how that changes with exercise. So we have some exciting new data coming in actually as we speak. Last week we heard from a South African group new updates. So we are really working on this project. When we looked at the skeletal muscle sections, we noticed that some areas that were strange to us at first place. So we asked our local pathologists to help out with the interpretation of this. What we did is we scored for atrophic fibers, which were more abundant in the patients and also increased a little bit with exercise. But also we saw fibers that are areas of fibers that were necrotic, and particularly there were already signs of necrotic fibers, sometimes large, sometimes small, in the patients before we actually did the PEM induction. We also spotted that not only the muscle damage was more, but also the recovery, the regeneration of skeletal muscle was also changed or different between the groups. And that's something we see in the bottom part here. Some people have asked us what these percentages are, why not everyone has suffered from these conditions, and that's actually something important to mention. Is that what we do is we sometimes have a large area to look for, and sometimes the area is very small, so we cannot distinguish between a very big muscle fiber that we look a big muscle area to look at and a very small area to look at, and even if there's one or two fibers that are different, then we call it a positive sample, so to say, or the pathology calls it a positive sample. That's what we see here. We also spotted there's some immune cell infiltration in the skeletal muscle and the T cells, your T cells, macrophages, and B cells, where we found differences between the two. And because of time, I will not go into a lot of detail here, but we are really interested in this link between immune cell function and skeletal muscle cells, and see how they interact with each other. So what we can conclude from this is that the lower exercise capacity in patients with long COVID, but also likely ME/CFS. We are conducting a study right now to see whether something similar happens also in patients with ME/CFS. Are likely completely different from the pathophysiology of post-exertional malaise. So we do know that patients that contributors to the reduced exercise capacity include the lower mitochondrial respiration or ATP production, metabolites that are related to fatigue were higher, and more glycolytic fibers. And that's something that you don't see changes in very quickly, essentially, in the skeletal muscle field. And also we spotted a lower power output per muscle cross-section area. On the other hand, we do see changes that are very rapidly occurring upon the induction of post-exertional malaise, just as that rapid local and systemic metabolic change that we see, not only in the blood, but also, so not only in muscle, but also in the blood. We saw some markers for muscle damage, but also recovery. So there is already a recovery process occurring as well. And maybe from previous PEM episodes, we don't know. And we do see some infiltration of certain immune cells that we now like to further quantify and identify which ones are they. Here we highlight mainly the T cells and the macrophages. And we also see some of these amyloid microclots occurring in the skeletal muscle. But again, we don't really know why they are there and what their function is. I did not show you this, but we didn't see specifically more N protein accumulation in skeletal muscle. It was not that there's more viral there were some viral remnants in skeletal muscle in patients as well as in controls, but that itself did not change so much and did not link to any of these parameters. So what are we doing right now? We are trying to summarize the field that is where people have worked on right now in the different parts of the world on the contributors of fatigue and the contributors of post-exertional malaise on the skeletal muscle level. So we've just written we're writing a review paper about this right now, and we're trying to summarize what other people have also seen and confirming our results, or sometimes seeing different things than what we see. Important critics was that's these average steps that are described already in the beginning. And interestingly, around the very same time as our publication came out, another PhD student in our lab, Moritz Eggerbusch, published a paper where we actually studied the effects of bed rest, strict bed rest on skeletal muscle function. So we were able to we're now able to link essentially the two projects together and to see really whether the deconditioning that people think have occurred in the patients is really the driver for the change that we see. And I can assure you that that's not the case. We are really trying to see separate phenotypes. You can also see that here already some of the patients are actually more active than some of the controls, whereas where all the patients suffered from PEM and none of the controls did. But we're doing a deep dive into these comparisons to really see the differences that are occurring in the post-viral diseases, including a new cohort of ME/CFS patients, and see how different they are from strict bed rest. So this is maybe interesting for you to see how these bed rest studies occur. They go in collaboration with NASA and ESA, the European Space Agency, and in the German Space Agency facilities. And there are people laying in bed for really 24/7. They have to have their shoulder on the bed every minute essentially of their time there, and 60 days long they are being trolleyed around the facilities to undergo MRI scans or muscle biopsies. And you see this is an image of our muscle biopsy procedures that we performed over there as part of that study. But I cannot show you the results yet. We are working very hard to finalize these final results. So what I can do show is that the take-home messages is that many patients with long COVID and ME/CFS suffer from post-exertional malaise, and that it really symptoms really worsens with exercise, physical exercise, but also mental exercise. We do like to highlight that exertional dyspnea or fatigue are really something else when we actually mean PEM. So that's something to take home, is that that these two things are really separate pathophysiological mechanisms, and I've highlighted some of contributors that are distinct between the reduced exercise capacity and the onset of PEM. We also know that exercise above a PEM threshold is going to be counterproductive for recovery. So above that threshold is likely not beneficial for recovery. We don't know what this threshold is and whether we can study that in more detail. We're doing this in more detail, studying this threshold in more detail now, but we do think it's patient and time dependent. I've also shown you that peripheral change in blood and muscle can help explain the fatigue and muscle pain, but we don't really know the underlying X factor, and that's something that people have suggested to me. So maybe I needed everyone who is a long COVID patient need to undergo muscle biopsy measurements. That's not the case, because we see some of these changes that I've just described with other diseases as well. So it's not very specific for long COVID or ME/CFS, and it makes it very difficult to turn this into a diagnostic marker, because we don't know the underlying X factor. If we would know that, that of course be a completely different situation. What we also what I can conclude now from the studies that we have already published is that strict bed rest really changes your skeletal muscle in a different way than long COVID and maybe also other post-viral diseases. So there are other contributors as well. Some of the practical implications that I want to highlight here, and that's something that because of this this seminar or this webinar was well viewed and will be well viewed and has already attracted a lot of questions. I was able to screen through the questions and see where I could already help with answering some of the questions that viewers have. So the practical implication is really that patients are encouraged to exercise below the threshold. We do know that exercise to some extent is good, because if you're completely bedridden, that can also in itself lead to deconditioning symptoms. So of course we encourage patients to do low intensive exercise, but the issue is a little bit that we don't know what kind of level of exercise is needed to avoid the PEM, and that's something that on a patient-to-patient basis you need to figure out essentially what is safe and what is not. We now try to find out different ways with pacing strategies and whether heart rate variability is a useful marker to help patients in deciding which intensities are safe. I got a lot of questions based on medications, what is helping them, what is not. I'm not a medical physician and I cannot answer those questions specifically, but I do have some citizen science skills, so to say, on social media. And this is actually an ongoing poll right now on Twitter, on X, as we call it now, about mast cell inhibitor drugs. We hear from a lot of patients that these mast cell inhibitor drugs, they can improve symptoms. And that's something that I wanted to hear essentially from patients who are taking these drugs to see whether that increases their PEM threshold or shorten that duration. And we do see that in 2/3. This is actually really today's results. That two-thirds of the patients who did give up did answer one of the three options, that it became less. But also we have some patients that where actually these drugs are worsening their PEM symptoms. So this is really something to highlight, that is not a one drug fits everything. But this is really specific, and we're trying to figure out from different perspectives which patients are likely benefiting from certain drugs. For that, we do need a big biobank, and that's something. The last thing I want to mention here is that we now are developing a Netherlands ME CFS cohort and biobank consortium. And that biobank is actually ongoing right now. It's a patient-initiated cohort and a consortium with lots of research in the Netherlands based on these on this cohort and the ME CFS disease population. It runs for 8 years, started last year, and it's harmonized with the UK biobanks and others worldwide. And we're aiming to include a lot of different types of participants to really get a good idea about the different certification for patients and severity, so that we can distinguish the bedbound and the housebound from the patients that have a higher threshold essentially for PEM. And then we're really great that we're able to get the ex-Solve colleague Kate Murray to be part of this project. So with that, I would really like to thank all the people that are part of the team, particularly Braden, who was able to continue this work based on this Solve M.E grant that we received, the clinical partners at our local hospital, and you see lots of different funding agencies that have really nicely supported our work so far. And I would like to thank you for your attention.
Thank you very much, Rob. This was an excellent presentation and a really good science. You answered many questions, but you also open up new questions and areas for research. So that's great science. There are certainly a lot of questions that came already, and quite a few during the presentation as well. So I'd like to ask a few questions, but maybe to start with, the diagnostic part, because as you said at the beginning, as patients, we know how to recognize PEM, but it is really, there's no objective diagnostic tool as of yet. Can any of the work that you've done be translated into a diagnostic test? Maybe you can say a few words about what a biopsy is. Is that something that could be used? I mean, it's an invasive procedure, but can you see something that becomes a diagnostic tool as a result of your studies so far?
Yeah, that's a very good question. So the biopsy is taken under local anesthetic, so you don't really feel anything essentially if you undergo this procedure, and it's super quick, so it's within a few seconds. I get a lot of questions from patients that want to be referred to our hospital to take a muscle biopsy, for instance, and want to get their muscle biopsy reanalyzed. This is something that is pretty difficult. As I mentioned also during the talk, we do these muscle biopsies for research purposes, and some of the things that we see overlaps with other diseases too, and that makes it difficult to say that based on one phenotype you can diagnose long COVID patients. So that's something that is not going to be feasible within the time frame that we are working with now. What we do want, of course, is to get an non-invasive assessment of skeletal muscle function, and that's actually something we're working towards now, is to get non-invasive markers that tell us something about the skeletal muscle, but not having to undergo all these procedures, so that it can be part of a clinical diagnostic tool, so to say. But unfortunately, we're not there yet to get it up and running super quickly. But that's at least something we're working towards too. Until we have a very clear idea what the disease is caused by, that determines also the diagnostic criteria. So if the disease is caused by something in the blood or in the immune system, or maybe in the gut, that determines essentially where the next steps are going to be based on what diagnostic marker is the best to diagnose that particular change, essentially. I do think, however, that it's not a skeletal muscle disease, and that's something that is important to realize, particularly because the PEM can also be triggered by cognitive or mental exertion, and that's actually something that people have criticized me on. That it's not specifically a skeletal muscle problem, but we never we never argue that is a skeletal muscle disease in itself.
Yeah, that makes a lot of sense. And there's been a lot of interest over the years in the ME/CFS community about the role of lactic acid or lactate. And of course, you've identified some mitochondrial dysfunction. Can you correlate some of your findings with levels of lactic acid? Do you think this is part of the metabolic dysfunction that's happening? Is this a cause? Is this an effect? How do you view that in the context of your work so far?
Yeah, that's a super good question. So what we see in our biopsies that we take at rest, not during exercise, we take them during rest, is what we see is that the lactic acid is not so much different in the muscle. But that's also what you can imagine is happening, because this is the muscle is at rest, is not in a stressful situation as you would during exercise, for instance. What normally exercise physiologists do is they take the lactate in the blood at maximal exercise. And again, there you would not expect so much differences between the groups, because both people went to the maximal ability of their exercise capacity. So also there, everything will be maxed out, so to say. It's interesting that at where in that scale the lactate starts to accumulate more, and with blood gas analysis, with a mask and breathing exercises, you can get an idea about the level at where this occurs. And what we do see is that it occurs at an earlier stage in the exercise test in patients with long COVID. So we do think there is a more tendency that their muscle starts to acidify quicker and lactate accumulates quicker. But it's really difficult to do this at rest or during maximal exercise. You want to do this in the middle somewhere to get an idea about the applicability of lactate.
Yeah. So in that model, would you view pacing as a way to kind of work below that threshold, or is pacing really helping the recovery? How do you see the role of pacing in managing time?
I think the initial idea is that you want to avoid overexertion. And if there is overexertion happening, that you quickly can adapt to that new situation, that you can sort of that you don't continue in the PEM for a long period of time, so that you try to shorten it. I've heard from patients that they can sort of feel it's coming up and then quickly adjusting the daily life activities to avoid that it continues for a long period of time. So the important thing is that we think is pacing. It allows people to better manage their threshold, so to say. And also, of course, when they are in the threshold, when they go over the threshold, that they quickly can adjust that. So we're hoping now that with heart rate variability measurements, we're doing some testing with that, that can give us an indication of when people are going over that threshold, and that they need to pace themselves back to resting values and shorten the duration of the PEM.
Yeah, that would be really interesting to see if those tools can help, because those are now available and there are apps that can help with that. So yeah, that would be really interesting. Rob, I want to go back to kind of the more molecular mechanistic. You certainly found evidence for mitochondrial dysfunction. Last year, there was a very interesting paper that came out of the NIH by Dr. Wang, who identified a specific protein called Wüst3 that seems to be overproduced in cases that are linked to ME/CFS and PEM. Have you been able to look at that particular protein? Do you think that could be part of the explanation? Is your research can address that as well?
Yeah, that's a very good question. Of course, we are super eager to dive more into that particular aspect. We do see, however, that the mitochondrial density, or one of the enzyme activities is relatively constant, but they cannot produce enough energy. So we do think there is an intrinsic problem with the mitochondrial ATP production, and that can be explained by this. That could be, in theory, explained by this particular protein. as I mentioned before, the size of the biopsy is only the size of a grain of rice. So with that, we can do a lot of measurements, but we cannot do everything. And of course, what we would like to do is to see whether that protein is also more abundant in our tissue compared to our controls, for instance, patients versus controls. But unfortunately, we're dealing with the scarcity of the tissue of patients that we get, and then we have to make really tough decisions to go for either this particular experiment or that particular experiment. And of course, we don't want to take another 100 biopsies from patients, although they're willing to come to our lab. But we have to juggle essentially with picking the right experiments at the right time to do. So it would be really nice to do this, and it would certainly be on my wish list. But there are lots of other things that I would like to have on my wish list, and that only goes to I can only do 2 things a day, or a few things a day.
Okay, so that's definitely part of future studies. Well, we talked about this as a post-infection disease, or really the outcome of a post-infection disease. How do you see the connection? You alluded to that maybe towards the end of the presentation, the connection to the immune system. What would be the cascade of events from a viral infection or infection in general, ultimate to leading to a mitochondrial dysfunction? What could be the connection? Is it something that develops over time, or is it acute? What evidence do you have for that?
So our studies or the work that we do is not directly linked to understanding the earlier onset of the disease, so who gets the disease and who does not. But we do know that from our long COVID cohort that there was a very specific moment that the disease started, which was the COVID infection or the SARS-CoV-2 infection. So we do think that is the starting point for this particular disease. And if you look at ME/CFS patients, they often don't exactly know when the starting point was of the disease. It's always around a rough period of time where maybe there was a viral infection. But I think the COVID pandemic taught us something that the viral infections can be a good starting point to understand the earlier onset of the disease. And people have come up with different types of hypotheses of viral persistence. So there are some viral remnants in the body that can trigger the immune system constantly, or as people have suggested things in the microbiome maybe that happen. So there are various hypotheses going on. An intriguing one is the autoimmunity, because that would explain also some symptoms that patients are suffering from, and that's actually something that with in collaboration with other people we are looking there into that particular aspect a little bit more. But it's a complicated field, to be honest, and I'm not immunologist, and that makes it also even more difficult to say something really specific about the autoimmune component of the disease and whether that is the most likely contribution of the disease. I don't know.
Yeah. No, that's that of course is something that we're all interested in identifying. And there was a.
Can I quickly interrupt? Can I quickly here? Because even there are of course other autoimmune diseases, and I think one of the issues, of course, with what we have in the field is that yes, we want to know the starting point, but for patients we also want to know how to deal with it and know what to do about it. And of course, that if it would be an autoimmune disease, we would have different types of options than comparing it with when it would be another type of starting point, so to say. In any conditions, it's not an easy fix, so to say, because otherwise we would have got the easy solution already if it would be something to do with blood pressure or with diabetes or with the standard thing. So it's going to be a tricky one to figure out what is the cause of the disease, and maybe there are also different people where different contributing factors are underlying, and therefore the treatment options may also be different from different patient populations. So that makes it even more difficult, this disease, to study.
Yeah. Yeah, no, that's we I think we certainly agree with that. And on that topic, you mentioned some of the things that you hear from the participants in your study about treatments that they use. There were quite a few questions from the audience about potentially other nutritional ways or supplements. Do you hear from people who have been using any particular compounds like that that seem to be working for some people? Maybe you could share some thoughts about what those might be, ways to supplement the mitochondrial function or anything else that could be helpful in that context.
Yeah, yeah. That as I mentioned also during the presentation, I'm not a medical physician and I cannot answer those specific one-to-one but I do also read literature and also hear patient comments and patient feedback, and that makes based on that general knowledge, so to say, that is out there in the open digital world, so to say. Some people have suggested that creatine supplementation is interesting for getting more energy, or carnitine supplementation that have been suggested by some people. The efficacy is always tricky to study. There have been some studies that have seen some slight improvement, others have not seen it to the same extent. But maybe also the patient populations are different, and I think this is really something on a person-to-person basis that it's worth trying out, I would say. I've also heard people mentioning that certain foods are worsening the symptoms, and those, of course, would be also ones that you could take out of your diet to be able to not get the negative sides of effects. But again, the details there are really...
That's interesting. And there was also a question or even a suggestion of there was a suggestion of maybe even intermittent fasting that could be one way that some people deal with that. So that's also affecting the metabolic balance for sure.
Yes, it does give a little bit of stress on the metabolic system, and that allows the body to sort of recover. Okay, let me say it this way. It may help with some symptoms, but it will not solve the problem. And that's, right, I think, the bottom line also with a lot of these supplemental and nutritional compounds. I've not heard yet anyone completely recovering from these symptoms based on a nutritional supplement or changing a diet, and that makes a search for a medical treatment, of course, more important.
Yeah. Well, thank you very much. Before we close, any thoughts about your future, or if you could share what are the other studies that you're working on now that we'll be looking forward to hearing from you in the coming days?
So we are, of course, working on this project where we combine the information that we get from our bed rest measurements together with ESA and NASA to link that up with our disease populations to see whether they are completely overlapping or not. So that's the first thing that we're currently working on. This heart rate variability is a side project that we're really interested in, because it could be an easy way for patients to track their performance and to track their daily life functioning, so to say. So those are the two things, and we also look for there are lots of things happening in the lab right now, but we really try to dive deeper into the skeletal muscle abnormalities that we see, linking with the immune system. So lots of things to do. So there's never a shortage of ideas in that sense.
Fantastic. Well, thank you so much for your work so far. We look forward to hearing more about the ongoing research. And Carmen, back to you.
Thanks, thanks, Oved, and thanks, Rob, for that presentation. I just wanted to remind everyone that our next webinar will be the first installment in a 4-part series on severe ME. And Solve is co-hosting this with our friends at the Bateman Horn Center. You can see the a little slide there about that. And if you wouldn't mind advancing one more, the first installment is going to be on the 9th. And I'll drop the registration link here into the chat if you'd like to sign up. You can sign up for one or more of the events in that series at that link. And again, it's on severe ME, care, rights, and research. We're really looking forward to this one. So I hope you'll be able to join us. We've got a lot of folks already signed up. And then I just also wanted to remind everyone, if you'd advance again, that you know, all of this that we do, these free educational webinars, our research and advocacy educational program, is only possible with donor support. And we're always leveraging donor contributions into bigger gains for cures and treatments for ME, long COVID, and other infection-associated chronic conditions and illnesses. So if you'd like to support our work today with a gift, we'd really appreciate it. You can go to solveme.org/donate, or you can use the QR code here, and I'll drop a donate link into our chat as well. You're going to get an email at the end of this event with a link to a recording of today's session, and we're going to post the event, the recording of the event, to all of our social media accounts. So keep an eye out for it there. Usually we'll put them up within 24 to 48 hours. And then just one last thing before, if you have a moment, when this event ends, you're going to see a brief survey that'll pop up on your screen. If you have a moment to share and you'd like to give some feedback to us on our events or anything else related to Solve, we'd love to hear from you. So that's it for us here. Many, many thanks again to Dr. Wüst, to Oved, and to all of you who attended today. Have a great rest of your day. Thank you so much.
Thank you.